And with this, I'd like to move on to the next part of our event. We're going to talk about research interest groups. We will have the faculty leaders of each interest group come up and tell a few words about what they've been doing last year and what their plans for the upcoming year are. All right, the first group. Oh.
So I'm going to stand in just for a very brief introduction for the Genes and Society. This is the newest rig, which has been in place for maybe 2 weeks. And so there is nothing to report from the previous year. They will have an amazing report next year's retreat. And the overall goal of this research interest group is to foster interdisciplinary collaboration among geneticists, social scientists, ethicists, and science communicators to address the societal implications of genetic research. And so this is open again to everyone across the university and within the community. And Martha Burford-Reiskin is one of the co-leads on that. And if you want to join this rig, you can go to the web page for the GGA. Her email address is also up here. And with that, I'm going to ask, as the other rig leaders come up to present, just advance the next slide and it'll tell you who the next speaker is. So back to Anna.
I'm one of the faculty co-leaders of the intrinsic group. I always forget what intrinsic stands for, but I think it's Initiative for Translational, Integrative, and Synthetic Plant Biology. Basically, we're a group of plant-focused researchers that get together weekly, every Friday. It's a lot of work to organize weekly meetings. So over the years, we started with faculty organizing these meetings. We've been running this since 2005. And so we've recently acquired the sponsorship of Genetics and Genomics Academy and the Plant Science Initiative. We've had now student and postdoc groups leading these events, organizing the events. This past year, we've held over 30 ideation sessions. We've had students present and postdocs present for most of those group meetings, where trainees present what they're working on currently, maybe some challenges they're encountering, some ideas for future work. This idea is to give the students and other trainees to experience the stage in a friendly setting, where instead of waiting until the end to ask questions, we actually interrupt the speakers after literally every slide and ask questions. This teaches students and postdocs to handle questions well, to see the types of questions that may come up when they go to more formal events, such as symposiums or seminars or conferences. We also had faculty give talks on things that they're thinking about, potential grant proposals that they're writing, potential papers they're trying to finalize. We also had a few external speakers, both corporate and academic people, talking about the exciting things that are happening in the realms of their companies or their institutions. Last year's organizers were Mario, Katie, and Steph. They are from Alonso Stepanova Lab and Joe Gage Lab. I'm very grateful for them. They've done a great job organizing the events last year. This coming year, our new leaders are 3 graduate students. Hannah from Ruben Rian Alvarez's lab, Jonathan Zirkel from the lab of Amanda Holzkamp, and Lucas Bauer from Colleen Doherty's lab. They have their first event that they're organizing this coming Friday. Again, the goals of this events, of this weekly Friday meetings, is to help trainees improve their presentation skills, provide various types of constructive criticisms for people that are coming up with new projects for grant proposals. We also invite local company representatives for us to learn what kind of services they offer, and also for them to see what kind of people we train, so that in the future, when our students and our postdocs are applying for jobs, those connections are already established. Also, if there's a new faculty or a new postdoc joining a group, we have those people come and present what their expertise is. So we are well aware of what's happening in other plant labs. And historically, we've been able to spark multiple collaborations, and that resulted in cumulative number, like I would say, over 10 collaborative grants over the years. And now we're moving to the epistasis group.
Second time I get impromptu called to stage. David must have gotten lost. We started this rig with David Rasmussen and Fred Gould out of an interest to predict genetic interactions, or basically the effect that genetic interactions or epistatic interactions can have on our understanding of evolution and applications of, for example, protein, all the way from proteins to organisms. We essentially met all throughout last year, maybe starting in November. It was a way to form community around reading papers that were of interest, and we developed a number of hot topics that were connecting our research and interests. Again, from people that were thinking about biochemistry and biophysics, all the way to people that might be interested in ecological interactions and kind of all of those under the umbrella of genetic interactions. Here are some of the questions that we had. To what extent do we need to account for epistasis when predicting phenotypes? Or is epistasis uniform across the genome? Does it tend to arise within modules, similar, regulating similar cellular processes, and so on? Our intention was to identify some of these topics that then would become NSF or NIH grants, specifically maybe we're targeting or thinking about targeting a BII proposal, and we're still sorry, David is here. Giving we're trying to build a single through line that connects our research interests, not necessarily shoehorn our current research programs into a proposal, but rather really come up with a new topic that might be kind of valuable for this BII kind of initiative, or this BII type of call for opportunity. I forget the name of the NSF thing. We've also been toying around the possibility of putting together a research training grant. I think that, or we think that epistasis would be a good topic that would be very broadly applicable throughout the GGA. So if you're interested in talking about that type of opportunity, please contact us. We intend to continue to meet in this kind of relaxed journal club style that would kind of foster these kind of questions and the development of these ideas for grant writing. And that's it. Oh my God, I'm still there. Sorry. Thank you.
Hi, everyone. I'm Katie Heil. So the Evolutionary Genetics Discussion Group, or EvoGen, my co-leaders, Raphael and Christina, we started this a few years ago, and we kind of have three main goals, which are to build community centered around common interest in evolutionary genetics research, to help trainees have opportunities to present their research in a more full-length talk format, and to highlight and promote evolutionary genetics on campus and beyond. We mostly run this as a seminar series. It's every Thursday on around at 4 o'clock. It's in Thomas Hall. We again mostly try to have students and postdocs present their research. This last year, we also had talks from a number of our new faculty and host social events. And this year, we just submitted a paper that was led by grad students across 4 different labs that are involved in this rig. And we hosted our first external speaker, Dr. Pengyao Zhang from Arizona State University. And to me, one of the highlights was that she really commented on how impressed she was with our evolutionary genetics community, which was our main goal. So this coming year, our seminars start on Thursday, September 19th. We have a really nice slate of students and postdocs giving talks this fall. And if you're interested, please send me an email and I'll add you to the listserv. And stay tuned. We're having another external speaker, Dr. Erica Larson, who works on speciation, and she's going to be coming in February of next year.
Hello. So together with Claire Gordy and Martha Burford Reisken, we have formed the Genetics and Genomics Education Rig. We're a little bit older than the Genetics and Society rig by about 2 weeks. So we started in July, I believe. So we do not have anything. Oh, we do have one thing to report. We did host a syllabus writing retreat. That was our first event where instructors came together for a morning of coffee and bagels and community to start working on our fall semester materials. But the goal of this rig is to bring together those who are interested in exploring evidence based strategies for teaching and learning in genetics education. So we're interested in having anyone who's interested in education join our group, faculty, staff, students, postdocs. And importantly, we really want to emphasize that you do not need to have any sort of background in education, just an interest. And we would be happy to have you come and hang out with us. So my email is there if you would like to join this rig.
Hi everybody, my name is Fikret Isik with GKI Jiang from Animal Science. We have been leading the plant and animal breeding research interest groups for a number of years now. And so GKI is a new rig leader. And I was in South Africa last year on sabbatical. So we were not as active, but we are planning to have more activities and um, have some seminars and meetings this year. Um, so one of the activities we organized was panel discussions about threshold models of quantitative genetics and how it's related to those binary traits and how we analyze and make selection decisions for binary traits. And the panelists were Chris Malteka from Animal Science, Dahlia Nielsen, James Holland, and Jikai Jian. So it was well attended. And, we offered lunch. That was one of the, I guess, the highlights. So up for this semester and next year, so one of the activities we are working on is to um, have that computing skills in plant breeding workshop. This is going to be in December. It's going to be taught by teams, different teams on different topics. It's going to be hands-on. How do we use different types of data, phenomics, genomics data for plant animal breeding? So it's the registration is open. You are welcome to visit the website and sign. Um, and then we would like to invite maybe a couple of speakers next year related to animal and plant breeding. So if you want to join our research interest group, just Google the name of the rig, and you will see my contact information. We'll be happy to add you to the email list.
Hello, hello. I'm Carlos Scholar, he, him, and along with Justin Whitehill, we get to talk about technologies and look at what new equipment, instruments, techniques are available on campus and beyond. So, this is the Genetic and Genome Engineering Emerging Technologies rig. We have Justin and I have uh, teamed up, and we both have interests in different technologies. So, I think we complement each other. And the goal here, our objective is to make sure we as a community, we're aware of the potential use of new instruments or new technologies on campus. So we started with Justin organizing over at the Plant Science Building an awesome workshop on laser capture with an instrument that's over there. And this was a one-day event, really uh, emphasizing training. We also had a workshop on or a seminar with a question and answer and demo session on Hamilton automation with the prep liquid handler in May. In July, we had Jeremy and and Danielle talk about the QIAGEN digital PCR platform that's different from droplet digital PCR. This is, if you go over there and talk to Jeremy, you can see some of the plates that this system uses and the QIA Acuity. And the goal here is to learn about what technologies we have and how can we use them together. So we plan on doing a couple more workshops and events this year. One of them will be with Oxford Nanopore Technologies, and I see Stuart and Austin over there, and they will help us with some of the bioinformatics. So Epitome Cloud and Epitome Desktop have improved over the last couple of months significantly, and now there are several workflows that we can tap into. We're working with Nanopore and the Research Facilitation Service to make sure that some of these workflows are available on the high performance computer cluster. Some of us have Opentron's robots and are not using them to their full potential. So we're going to try to do a workshop on the Opentron and Opentron Flex to get someone here, a human being, to teach us how to use the robot and do a better job with it. Matt over there and Illumina, there are some new kits and new ways of doing library preparation so that you can submit high quality samples to Kelly and the GSL. So we will try to do a wet lab. A couple of years ago, we did a 1-day event where you would bring your samples and did the DNA flex. So we will try that. And we'll do something similar, hopefully, if I can convince. And I haven't told Stuart and Austin. So, Stuart and Austin, can we do an Oxford Nanopore Technologies library prep afternoon and maybe some QIAGEN kits and prep? So, these are just some options. You're doing the research and have the questions and may have a challenging technique or a challenging question, and if just email us and send us ideas for workshops. Thank you.
I'm Max Scott, co-lead this genetic pest management group with Fred. Fred's also in entomology, and with Alan, who's in mathematics. So genetic pest management, we use the pest itself as the control agent, which is kind of counterintuitive, right? You want to control something, you release more of the pest, but it does work. So we're a really interdisciplinary group with molecular genetics, population genetics, mathematics, but we also are active in the Genetic Engineering and Society Center and work with people like Zach Brown and Jason Delborn. Because, you know, really, if you're going to work in this field, really combine molecular genetics, genomics, mathematical modeling. You've got to be aware of the economics of what you're trying to develop, public outreach, public engagement. You can be stopped before you even get close to doing a field release. And it's really good to be aware of government policy in the area of biotech regulation. So we're very interdisciplinary. I think we had a pretty good year. We published some papers. We've got some new funding that'll provide continuing support for GGS students in entomology and mathematics. That's, of course, the challenge in GGS, to support the students after the first year. And, well, I think for a lot of us, highlight of the past year was the biannual Gordon Research Conference on Genetic Biocontrol, which is the main conference in our field. Fortunately, this year it was in Barcelona, which I'd never been to before, but really enjoyed it. It was a hotel near the Mediterranean. That's why we're out kayaking. So Ariel, I think just mentioned, got an award, travel award, and she used it to go to this conference. And she was awarded one of the 2 poster prizes, which was a big success, but people really liked her poster. And yeah, actually, there were a number of people from NC State at the meeting from multiple departments. And, you know, this was didn't go unnoticed. A number of people said to me, you know, NC State's really strong in this area, genetic pest management. And I would agree. And I think, you know, the reason we are, it's really Fred's vision, you know. I mean, before we even had a faculty cluster awards, Fred convinced the university to establish a center of excellence essentially in genetic pest management, which is why I came and Marseille came. And then with the Genetic Engineering Society Center, people became interested in the human public engagement and economics of genetic pest management. So I think it's a really strong rig in the university, and hopefully we can keep it going after Fred retires. So we're going to keep going at Journal Club. This is a really fast developing field with the widespread use of CRISPR-Cas9 technologies. It's a challenge for all of us to keep up with it. So we have a Journal Club that meets every 2 weeks. New members are welcome. We're not going to hit you up to present straight away. If you're interested, contact me. This year, I think Ariel is running it. Usually one person presents, but if a paper has a significant component, say, of molecular biology and mathematics, we might split it between 2 people to present each part of it. Conferences, we're going to, actually I fly out to Japan tomorrow to attend the Entomology Congress, which is going to be in Kyoto. There's quite a few talks in genetic pest management at that meeting. And the number of new projects starting with the new grants. And I'm sure we'll write more grants in the first year.
Hi, everybody. I'm Ben Callahan, and I'm presenting for the Microbiome Rig. I would argue that this became a rig as of this moment right now, so maybe the youngest one. So that being said, basically this is a through line from the microbiomes cluster, CFAP microbiomes cluster, and we're trying to understand what it means to be a rig in the next year, and we're trying to reach out more broadly to the community to get more involvement. As an established cluster, we had a variety of activities. Sort of one of the core activities that we do is we host a monthly seminar series called the Microbiome Monthly Meetup. And we have a couple invited speakers every year. We host different labs from around the Triangle area. And then we also host trainees as part of that. We also have a cluster webpage, which is kind of the central resource for this rig at this point. And you can get the seminar series information there. I think we just posted the New Year's speakers just now. In the last year, we had a couple nice sort of individual accomplishments, a distinguished professor, a couple university faculty scholars, a national award from the American Society for Microbiology. Nathan Crook was awarded NIH New Investigator DP2 award. Maybe the biggest thing, though, was Christine Hawks, in collaboration with a couple members of our cluster and other members of GGA, was awarded a new training grant. And so we just have our first cohort of students that we're onboarding and they're starting out right now. So that's very exciting. And we also last year developed a strategic plan document. That was us a cluster. That's something we might revisit in a couple of years as we try and become a rig that broadens beyond the initial cluster. So for the next year, again, our core organizing is around this Microbiome Monthly Meetup. We're going to be working on helping this first cohort of the training grant students succeed. But we'd like to expand our reach. We have a mailing list. If you'd like to be added to the mailing list, just reach out to Casey Terrio or myself or really anybody in the cluster. We have a number of new projects, like new funding, basically, on several topics: microbiome stewardship, transductomics, which is a metagenomic and metaproteomic methodology for being able to see transduction by viruses, sort of as it happens. A project on inactivating C. diff toxins, and ongoing projects on microbiome-mediated crop resilience. And actually, listening to the forestry talks, I sort of wonder to what extent the microbiome might play a role in forestry productivity as well. So please reach out if interested. We'd love to hear from you. Thank you.
All right, well, hello everyone. So my name is Nick Bushler, and I'm one of the co-leaders of the QCDB, or Quantitative and Computational Developmental Biology cluster. I want to point out that we're inclusive, and so actually half of our members, we were originally a CFAP cluster, we're actually cell biologists and not developmental biologists. So I think at some point we're going to change the computational to cell biology, because that's what we really are: quantitative, cell, and developmental biologists. So very similar to the microbiome, we were originally a cluster hired through the CFAP initiative, and we're a new rig now. There's been interest in this. And so our activities over the past year have really originally been as a CFAP cluster. And one of our marquee events, which I'm showing up here, is we have an annual retreat, a QCDB retreat. It's very well attended. We have 40 to 50 participants or more from across different colleges here at NC State who have interest in cell and developmental biology. We have short talks and poster presentations from trainees and also faculty. And this is something we've really enjoyed doing. And as you'll see in the next slide, we're doing it again. And I invite all of you to attend. It's open to everyone here. The other thing is we host seminar speakers very broadly, which I list some of here from the past year. We also foster sort of new initiatives to get multidisciplinary grants. This year has really been, the push has been with AI. Dr. Arguello Miranda, as you can see him, has been very productive here, sort of using AI either for image processing or mathematical modeling of cell biological processes. And we also host workshops. So this year we had a workshop that involved NC State researchers working with fungi and also Duke mycologists coming together to learn some of these tools for image processing. So that's something we like to do, sort of foster new relationships, have workshops in quantitative biology for the community, and have our marquee event, this annual retreat. And so moving forward, and that's very unfortunate, there's no image here. Oh well. What I did notice though, is you'll see in these rotating slides, you're going to see that there is a QR code. We have a keynote speaker this year. So this is happening next month. You're all invited. Anyone can register, scan that QR code, register yourself, and come attend. You can present a poster. You can be nominated for a short talk. We've got awards for trainees. We'll have great speakers. We have 3 local speakers, a keynote speaker, which will be Karen Kazaa from Columbia University. So I encourage you to attend. And we also continue with our workshops. We have an upcoming workshop in May. It's a computational workshop by Giulio Belmonte in the physics department, who really specializes in developing computational methods for modeling cytoskeleton in crawling cells, attached cells, thinking about a lot of the properties of multicellularity and cellular processes. It's sort of a week-long workshop where you learn how to use the software, and it's also a hackathon. So stay tuned for that. Again, it's open to the community. So thank you.